To stain up the bacteria, I'm going to put them all on one slide. I'm going to have the 3 different colonies I'm interested in. I'm going to make little smears of each colony. But so that I don't forget which is which, I'm going to write onto the frosted end of the slide a description of the colony. So I'm going to write large. I'm going to write yellow. That's colony number 2. And then the tiny small one, I'm just going to write small. I'm writing in pencil because the solutions for the stain, the gram stain, they wash off pen. I need to make 3 little blobs of water using sterile water, because we know that bacteria are in tap water, so we've got to use sterile water. And then I'm just going to gently touch the large colony with my stick. Just a little bit, you don't need much at all. Then I'm going to rub that into the water here. That's the large colony that I'm interested in. Okay, I'm going to do the same again now for the yellowy colony. Just gently touching it with my stick. I don't need much. And then that's going to be into the, I'm going to rub that into the second blob of water with a new stick every time. I'm just going to pick up one of the small colonies, rub that into that third little bit of water. So there I have 3 little smears of the 3 colonies I'm interested in. I'm going to stain them up. I'll show you how I do that. If you work in microbiology or study microbiology, you will know the gram stain. It is the bacterial stain that everyone uses. And the reason for that is that bacteria can be split into 2 categories. Those that stain purpley blacky colour with these 2 chemicals, and those that do not retain this pigment, and they end up staining pink with the saffronin counterstain. And the reason they do that is you have some bacteria which have a thick cell wall and some bacteria that have a thin cell wall. So if it has a thick cell wall, then it will be able to retain this pigment. If it has a thin cell wall, then it won't be able to retain this pigment and it will pick up the counterstain instead, so it'll be pink. So we got gram positive, come out purple black, gram negative, come out pinky colour. I'm going to show you how I do the staining technique, and this is standard for every lab you work in. You will need crystal violet, grams iodine. These 2 work together in the cell wall of the bacterium, and then we're going to wash away the excess crystal violet iodine complex using alcohol. Some people use acetone, we use alcohol. And then we're going to counterstain with saffronin. So we have our bacterial smears here. They wait for them to be dry. Now, you can't really see it on the camera, but there is a smear here and a smear here of the small cells. Okay, so what we do is we add crystal violet for 1 minute. After a minute, we're going to wash that off, and you can see some purple staining on there. Now we put the grams iodine on for a minute. Wash that off, and what you'll see is the cells have now turned black, and that's because the crystal violet and iodine have made a complex inside the peptidoglycan bacteria cell wall. And there we go, I'm just putting the paper underneath so you can see that it's now gone black. So now we're going to add the alcohol, and that's going to be sloshed on, and the crystal violet iodine will come out of the cells. Alcohol washes away the excess crystal violet. It removes it from any of the cells that have thin cell walls. These 2, can't really see them through the camera, but they are there. Okay, what I'm going to do now is I'm going to counterstain it, and that's going to be with the saffronin, and that's going to go on for a minute, and the saffronin will stain anything that has not got the crystal violet in. Right, so they've gone a little bit pinky as well, and they are ready to look at under the microscope. You can see some of the cells with the 400 magnification on the microscope, but the 1000 magnification is the best. And for that, you also need the oil. So that's an oil immersion. Let's find out what we've got. I zoom in at 1000 times magnification to find that my sneeze has 2 different species of gram-negative bacilli and a gram-positive coccus in streptococcus formation. My cough has the same bacteria. Have a great day.